Please welcome Dr. Kedalin Rusnak.
Hi everyone, thank you for having me here. So first of all, I'd like to make a correction. I don't actually have a PhD. I did work as a PhD student for a few years, but like many great founders, I'm a dropout. So a bit about myself. I was born here in the country with drive-through wine cellars. I hope the guests from abroad have the time to enjoy some of them. Then I moved to Austria to study biotechnology, then to Edinburgh to study bioinformatics. Then I came back to Austria, started my unfinished PhD in evolutionary biology in 2018, and that's how I started to work on a project that's now called RepliFactory. The motivation was that we wanted to study how bacteria adapt to stressors such as antibiotics and heavy metals, and we wanted to look at the genetic mutations that are responsible for this adaptation. And this is an example of such a mutation. Here we have a stop codon that appeared in a protein that is a cobalt channel in response to cobalt stress, which really makes sense. If there's too much cobalt, you don't want a hole in the membrane to let it through. And to make bacteria evolve these mutations, you run experiments like this, where you let bacteria grow, you hit them hard, wait until they start growing again, hit them even harder, wait until they start growing again, hit them harder, and so on. And that's a process that normally takes a lot of lab work, a lot of pipetting, a lot of measuring optical densities, measuring growth rates, a lot of cleaning, and of course, after all that hard work, a lot of coffee, because drinking Moldovan wine in the lab is usually against university policy. But luckily, most universities have automated coffee machines, automated dishwashers, and you see where this is going. We built an automated fluid handling robot. This is a schematic. It has these 7 culture vessels with integrated optical density sensors and stirring, and we need just 3 pumps to control the medium composition in all of them because we use these 7 pinch valves. And it looks like this. We use 3D printing to build a case which is very compact. It's not bigger than it has to be. And inside there is a little computer that runs all of the 18 motors in the device. And a big advantage over competitive systems is that we can detach the fluidics and autoclave it to sterilize it because it's made of materials resistant to high temperature. Here I have a demo of the graphical user interface. Yeah, thank you. You can press a button, have the valve open, and that means that when you run a pump, it's directing the flow of liquid only in that vial that is selected by the valve. We also have a magnetic stir that mixes the liquid and makes sure that the bacteria don't settle, and they're well oxygenated. And you can see the waste pump in the back spinning and aspirating the excess. And of course, the optical density sensor is also there. It's just a laser and a photodiode on the backside that measures how much light passes through, and you can tell apart vials with many bacteria from vials with fewer bacteria. Then there's a simulation environment that lets you see what the device thinks the experiment will look like. You can adjust a bunch of parameters to edit this simulation and the way the experiment will run. And if you're happy with everything, you run a bunch of self-checks that verify that all parameters are within acceptable limits. And then you can press the magic start button that starts the long-term evolution experiments. And of course, you can adjust the bottle volumes to reflect what you actually have on the bench. And when the experiment runs, you can monitor a bunch of metrics. It looks like this. You have stirring and OD measurements every 60 seconds. You can see a red light going through all the vials and a little bit of pumping. And after that, you can look at the data. and run a bunch of analytics on the data. Here we see that first the stress was a bit high, and then the optical density recovers because the bacteria adapted. And you can compare the replicate experiments between vials. You can compare them to older experiments and so on. Now, RepliFactory was built to study the evolution of bacteria, but the device itself has also evolved over time. It started many, many years ago. It looked as a prototype which is not very well polished. There were many iterations of every individual component. Especially during COVID, development was at home because there was not much else to do. I wasn't allowed to be in the lab very long. And some versions of the device had this unconventional hot glue attached lasers. And then we changed different versions of the PCBs, and in some cases we had to use this traditional blue tape to fix electrical mistakes. And at some point we started a small batch production, and then the batch increased, and I moved development to a place called Happy Lab, where I could use this very nice metal laser cutter among other cool instruments. And last year, this was actually above the capacity of Happy Lab, and I had to rent a dedicated office. And here you see the chief assembly technician, also known as Dad, and my three 3D printer employees, and the label printer. And we made met a quite tight deadline with a bunch of devices. It was 1/4 of a ton of devices. And here's where they went. In 2025, the by far largest customer was an organization called SSP. Back in 2020, it was just one prototype that they had. And we have a fantastic partnership. I'm really proud to have them as customers, but that's also a big risk. So now I'm trying to diversify the customer base, and hopefully in 5 years we see a continuation of this growth. And here are some pictures of users. We started in Barcelona in 2019. We had this little summer school, and then at the University of Tartu in Estonia. And these are the first external users at Indiana University in 2023. IST Austria is the place where I was doing my PhD, and kids really loved the device. And then in Purdue University at SSP 2024, also at NMSU, and in Georgetown. And in 2025, SSP expanded to 5 locations: Lehigh, Pacific University, Albion College, Guelph University. Yeah, that was it. And IMC Krems is where I also work now part-time. RepliFactory devices almost replaced that big bioreactor for a fraction of the cost. And the University of Vienna, Boğaziçi University in Istanbul, and the Armenian Bioinformatics Institute also started using our devices. And this slide is empty because I don't have any devices in Moldova, but I hope to change that, and I have a proposal for how to do that. I think Moldova would really benefit from a Moldovan research summer school where students do hands-on research experience, and they work on real science. So not like the high school science projects, like real scientific questions. And the living and learning community that it can be built around this is really transformative. There are 2 examples, SMTB and SSP, and many students refer to this summer experience as the best thing that ever happened to them in their life. And I hope to bring something like that in Moldova as well. Thank you.
Thank you so much, Cătălin. We are so proud that our people from Moldova make such